endotoxins, and a lot of other things for NELT3. Interestingly, for all of the plant people here, you might have recognized some of the domain architecture because these NLRs are very similar to the plant resistance genes. And that's because these knot-like receptors are a common theme in immunity. They're conserved between plants and animals and other lower organisms. And it's actually the inverse of what you see with a lot of proteins. Instead of being a few proteins in Drosophila and a lot in mammals, there's only about 22 in humans, and as you go to sea urchins, you get 200 or 300, and plants have hundreds of these R proteins as well. And so these all serve a similar function of sensing problems in the cell and producing a response. So it's usually gene expression, cytokine release, and cell death. And you can see this with the plant genes and with the mammalian genes. And this is a fundamental innate immune mechanism to recognize both danger-associated molecular patterns and pathogen-associated molecular patterns. So the organism has a lot of necrosis, the cells are going to sense this. And these are environmental stresses that plants may very well respond to with these genes as well. So back to NELT3, the mechanism through which it gets activated in our system is through a two-hit model to regulate the IL-1 beta. We treat the bone marrow-derived MAGs with the endotoxin LPS for 4 hours, and this upregulates levels of pro-IL-1 beta along with pro-IL-18 and NELT3. And after that, we give the cells a second hit, which actually triggers the NELT3 inflammasome. What I'm going to show you is with my favorite pore-forming toxin, SLO, a related protein toxin called titanolysinol, or TLO, and nityricin, which is actually just a potassium ionophore. And these activate the NELT3 through still yet undetermined mechanisms. NELT3 oligomerizes, binds to the adapter ASC, which recruits caspase-1. This causes caspase-1 to activate, and caspase-1 leads to cleavage of IL-1 beta and IL-18, as well as secretion, and it also leads to HMGB1 release and a form of cell death called pyroptosis. So HMGB1 is another protein that gets released. This actually has a day job in the nucleus of cells as an abundant non-histone DNA binding protein. So it just kind of sits there, does its transcriptional thing. But once it gets out of the cell is when it becomes really exciting to me because it has a major role in sterile inflammation. So it binds to a couple receptors, TLR4 and RAGE, and it's actually involved in late-phase toxicity, in physical injury, and a whole host of inflammatory diseases, such that you can give mice a lethal dose of LPS, and if you give them neutralizing anti-HMGB1 antibodies 4, 5, 6 hours later, the mice will survive the lethal LPS dose, because this is a late-phase toxicity. It's also been implicated in lupus, cystic fibrosis, inflammatory diseases, cancer, and a whole host of other things. So structurally, it's very positively charged, and then it's got a negatively charged tail that helps keep everything together. HMGB1 is known to be released by the NELT3 inflammasome, as I said. It actually also binds in vitro to IL-1 beta, and it turns out when it does this, it can actually potentiate the effects of recombinant IL-1 beta. So HMGB1 can work with IL-1 and give a synergistic result for inflammation. So we entered this research with a few hypotheses. One is that the cells respond to SLO similar to other pyrogens, and that SLO will mediate both HMGB1 and IL-1 beta release, and that this release is actually a common pathway. HMGB1's day job is in the nucleus, so we hypothesized that the way that HMGB1 gets out of the nucleus is you probably have a nuclease freeing it. And if this is a common pathway, this leads us to the surprising prediction that the cytosolic IL-1 beta, this release, might also be regulated by nuclease. So we know that toxins promote NLRP3-dependent release of HMGB1. We can do that through immunofluorescence, where we stain cells with DAPI in red, HMGB1 in green. We give them SLO for up to 30 minutes, and we see that we can see visually the HMGB1 leaving the nucleus of these cells. We can quantitate this in graph, and we see that it's NELT3 dependent, in that if we take macrophages from NELT3 knockout mice, we see that there's a decrease in the amount of HMGB1 leaving the cells. We can confirm that this is leaving into the extracellular supernatant, because if we treat cells with TLO, we see that in wild-type but not NELT3 cells, sublytic doses promote HMGB1 egress. If we block NELT3, or if we use caspase-1 deficient mice, or if we treat with a small molecule inhibitor of caspase-1, we can block the TLO-induced release. If we crank the TLO up to lytic doses, we're getting it coming out generally. So we know that this is a regulated method. So the first question is HMGB1 required for IL-1 beta release? And we actually took advantage of some collaborators had some neat knockout mice where they had HMGB1 floxed out and they crossed them to a CD11C Cree mouse. So these are mice where their dendritic cells, which are CD11C positive, lack HMGB1. And dendritic cells behave similarly to macrophages as far as inflammasome things go. And so what we found is that IL-1 beta processing was impaired in these HMGB1 knockout DCs when they were treated with nigiricin or SLO. And it's quantitated in this western blot here. We see a big decrease. Now, if you're astute, you'll notice that these HMGB1 knockout DCs also have a fair amount of HMGB1. And the reason for this is HMGB1 is a very abundant protein. It's about 1 million molecules per cell. And our dendritic cell cultures are not pure CD11C positive cells. So we actually sorted the cells to sort out the CD11C positive cells from the CD11C negative cells. And I asked, is this CD11C positive population mediating the release, or is the CD11C negative population responsible for it? And what we see is the CD11C negative cells don't produce any IL-1 beta at all. And the HMGB1 knockout cells, again, only produce about half as much IL-1 as the wild-type cells. So this shows that IL-1 beta release requires HMGB1. So we know that in apoptosis, HMGB1 can be released under certain circumstances, is released under certain circumstances, and the DNase gamma inhibitor will block HMGB1 release in apoptosis. So we asked, is HMGB1 inhibited by this inhibitor in our system? So the DNase gamma inhibitor is F-methoxycyclohexylalanine, which is going to be shown here as FCA. And what we see is if we treat the cells with this inhibitor of DNase gamma, we block HMGB1 egress from the nucleus. Interestingly enough, we also completely inhibit IL-1 beta production in these cells that we've treated with this FCA. And it's similar to the caspase-1 inhibitor, YVET. And this occurs following both streptolysin and nigiricin treatment. If we look by Western blot, we can see that this is actually not just blocking mature IL-1 beta release, but also mature IL-1 beta secretion, processing, so that caspase-1 is not apparently cleaving the pro-IL-1. And you can see that the pro-IL-1 levels are increased in the inhibited cells over the wild type, and concomitantly there's no mature IL-1 beta being released by the cells. The FCA does block HMGB1 release, though this is not as robust as IL-1 beta. This is because the cells are dying and releasing HMGB1 once they've died. To also check whether this is just non-specific nuclease activity or if other nucleases play a role, we looked at a panel of nuclease inhibitors. So a second DNase gamma inhibitor, Ponicil Violet, blocks HMGB1 IL-1 beta secretion, just like FCA. ATA is an inhibitor of protein nuclease interactions that also blocks IL-1 beta release. If we use topoisomerase 2 inhibitors, toposide or genistein, we see that these do not have nearly the same inhibitory capacity that the FCA does. One further assay of confirming this, and also asking, does this hold up in the mouse system, does it hold up in human cells, is we can use siRNA with THP1 cells. So THP1 cells we can transfect with the DNase gamma siRNA, and we differentiate them into macrophages with PMA for 2 days. And then we treated them with nigiricin for 30 minutes and asked, are they producing as much mature IL-1 beta? And what you can see is when we treat the cells with the siRNA, we block the amount of mature IL-1 beta coming out, and we reduce the amount of HMGB1 coming out, just like we see with the FCA. So this shows that the FCA is specific, and that this is DNase gamma that is responsible for this phenotype that we're seeing. So one other question is this restricted to the NELT3 inflammasome, or is it seen in other inflammasomes? And so Salmonella actually, in fact, activates the inflammasome, and it activates a different protein, not NLRP3, but instead NLRC4, also formerly known as IPAF. And so if we treat cells with Salmonella, we get IL-1 beta release, and if we treat with non-invasive E. coli as a control, we see that they don't really promote a whole lot of IL-1 beta production. If we treat with the inhibitors, we see that this blocks, it cuts down the amount of IL-1 in mating response to Salmonella. And if we look by Western blot, once again, it blocks mature IL-1 beta processing. So following salmonella infection. So the question is, if IL-1 beta is not being cleaved and released, what's happening? So we looked to see if caspase-1 was active in these cells we treated with the DNase gamma inhibitor. So here we took the LPS prime bone marrow max, and we treated them with a reagent called FLICA, which is a fluorescent caspase-1 substrate inhibitor, so it will irreversibly bind to active caspase-1. And when you treat cells with SLO and this FLICA reagent, you see that the FLICA localizes to small punctate structures near the nucleus. And these are also co-stained with the adaptor molecule ASC, which is shown here in red. So you can see these in yellow. And when you treat the cells with the FCA, you actually block this ASC and this caspase-1 recruitment. Importantly, these are also the cells that lose HMGB1, because we've stained these same cells with HMGB1. So the cells that don't lose HMGB1 are also not the cells that are undergoing this inflammasome recognition. So these cells show a little bit of cytosolic caspase cleavage, but the FCA blocks this punctate structure formation. And it also blocks HMGB1 transmission, we've shown in other assays. We can also ask, does this happen biochemically? So if we take the cells, and if we look for caspase-1, we can see that the proform is formed normally, and if we treat cells with SLO, they make the cells cleave caspase-1 to form the P20. In the presence of either the FCA or the ponosyl violet, which are both DNase gamma inhibitors, we completely block this caspase-1 activation. So we've shown that the cytokine processing is completely inhibited by this DNase gamma inhibitor, and so this is a fundamental mechanism for the inflammation. The other main thing I told you about, in addition to IL-1 beta and HMGB1 release, was cell death through pyroptosis. So pyroptosis is a form of programmed cell death. It's similar but distinct from apoptosis. It's most clearly defined as being caspase-1 dependent. It's an inflammatory cell death, and to make things more confusing, it's caspase-1 dependent, but it doesn't require caspase-1 cleavage. So caspase-1 gets activated in some manner, but figuring out exactly what's going on is still ongoing. So a couple big things. Pyroptosis, the cell looks like it's bursting, similar to necrosis. It's not packaging itself into membrane blebs. It's also an inflammatory cell death. So whereas apoptosis is tolerogenic immunologically, pyroptosis is pro-inflammatory. So we can look in macrophages at pyroptosis using similar assays to what we're looking at in the normal cells. So if we treat the cells with an increasing dose of streptolysinol, and we can measure the amount of propitium iodide the cells take up. So as the cells get killed at 5 minutes, we see toxicity increasing. And 5 minutes is, the 5-minute toxicity is direct toxicity due to SLO. And the good thing we know is that there's no difference in the binding of SLO in our knockout macrophages that are deficient either in caspase-1 or in NELP3. So at 30 minutes, we see that these B6 macrophages, which resisted incredible amounts of toxin, most cultured cells, like the CHO cells, they start dying about here. And if we give them 30 minutes, suddenly the cells that were resistant are now extremely sensitive. So the number of cells that are dying are way higher. And so this 30-minute death is due to programmed cell death, which is specifically pyroptosis. We know it's pyroptosis because if you look at those knockout macs, they don't show the same degree of cell death when we knock out the critical inflammasome components. So we know that this shift is due to pyroptosis. We can now ask, is DNase gamma necessary for pyroptosis? So we find out that the short answer is no. We see that in the cells in the trace I showed you before, the cells are very sensitive. When we add the DNase gamma inhibitor, we get maybe a 2-fold change, but this is not the 20 to 30-fold change that we see with the knockout mags, or if we inhibit the inflammasome with an inhibitor with the potassium chloride, which inhibits the inflammasome activation, we see still a very broad range. It's actually consistent with other studies showing that you get a little boost to pyroptosis when you actually have active caspase-1 fully cleaved. So this is entirely attributable to caspase-1, the failure of caspase-1 to be cleaved, even though it's being activated for pyroptosis, which we also saw with immunofluorescence. If you remember, you see a little bit of hazy green with the FCA inhibitor, and so this is cytosolic caspase-1 that is presumably not being cleaved, but being activated, and likely responsible for this pyroptosis. So in conclusion, I've shown that both the IL-1 beta and the HMGB1 release require DNase gamma inhibition actually blocks focus formation and caspase-1 cleavage. And the cytokine processing requires DNase gamma. So, interestingly, as I just said, DNase gamma inhibition does not block pyroptosis. So this lets us separate cell death from cytokine release. And this also helps us put HMGB1 in series with IL-1 beta for processing. HMGB1 is actually necessary for IL-1 beta processing. And in data I'm not showing you, we also know that HMGB1 associates with IL-1 beta following release. And so this is a novel therapeutic target because HMGB1 is extremely pro-inflammatory. IL-1 beta is also strongly pro-inflammatory. We can block both of them if we can target DNase gamma or this pathway that leads from DNase gamma to HMGB1 release. So this will have great therapeutic implications because we don't have to hit just one, hit the other. We can stop them before they get out of the cell. So, in conclusion from both parts, I hope I've shown you that streptolysin is a potent tool for understanding the cellular and immune processes. As the SLO is specifically sequestered and shed via blebs in normal cells, but that in immune cells like macrophages, they can respond to SLO through an ALK3-dependent mechanism that releases pro-inflammatory cytokines to alert the rest of the immune system, specifically IL-1 beta, HMGB1. We know that these both require DNase gamma, and DNase gamma is also needed for caspase-1 activation. Also, I've shown for the first time that IL-1 beta needs HMGB1 for processing and release. And that, as I said, targeting DNase gamma is going to give us a double hit against inflammation. So, the future directions I envision is that there's a number of different pathways open, and I've got a lot of interest in teasing the system apart further. So one question is, to combine both parts, what are the mechanisms of membrane repair in the macrophages? So when I showed you the pyroptosis, the macrophages can withstand about 10 to 20-fold more toxin than your average cell. So macrophages are a great tool for understanding the mechanism of membrane repair and how cells manage to successfully reseal. This is important not just for cells surviving perforin and other cytolytic molecules and surviving bacterial toxins, but every time you exercise, your muscles tear. And if your muscles have plasma membrane repair defects, you end up with muscular dystrophy. So understanding membrane repair also has potential application in other tissue types, and if we can understand it in macrophages, this may help apply to other systems. Interestingly, these MACs go from being tanks to just completely falling apart under pyroptosis. And so the cells need to dismantle their membrane repair machinery at some point to execute this pyroptosis. So the question is, how do the cells activate this switch? They go from being resistant to taking themselves apart. There's also a lot of details that I like to fill in with this DNase gamma pathway. So although I've shown you DNase gamma is necessary, we haven't sorted out how NLRs and PIHINs can activate DNase gamma, and exactly how DNase gamma releases HMGB1, and how this permits caspase-1 activation. Because caspase-1 is thought of being in the cytosol, DNase gamma presumably is in the nucleus, HMGB1 is definitely in the nucleus, and so this has to be some sort of trafficking interplay that allows this immune response to be generated. And so this is also a platform for understanding the common themes of innate immunity. Because as I said, plants have a lot of these R proteins. Other organisms share a lot of these proteins, and understanding what we can learn from the mammalian system, we can presumably translate to other systems, and this will help us design resistance mechanisms that will help protect plants, or will potentially also sensitize plants, because these pore-forming toxins have been a great tool, and I think the people that have worked with the cry toxins from Bacillus thuringiensis understand this, is that we've made BT corn, and this kills boll weevils and other parasites from basically just expressing a pore-forming toxin in a plant system. And so I'm also interested in developing novel therapies to target all of these pathways and turn this into therapeutic benefit. So this couldn't be done without a lot of people. I'd like to thank my current mentor, Russ Salter, members of